I'm going to be talking today about medulloblastoma since that's my studies. Medulloblastoma is the most common malignant pediatric brain tumors, and it occurs in 16 to 25% of cases, mostly common in ages of kids from 1 to 9. They are characterized by 4 subtypes: wingless, sonic hedgehog, group 3, and group 4. And it all depends on how they're characterized by their location, by their genetic signature, and their nodularity or the histology. Chemotherapeutic drugs are synthesized to target the origin of medulloblastomas, and this is partly actually of where my thesis comes in. So our lab studies the cell cycle regulatory protein named SPEEDY or SPY1, and this protein overrides the cell cycle checkpoints, which induces proliferation of the cells, even with DNA damage that comes from chemotherapeutic drugs. And also, SPY1 is implicated in the maintenance and the expansion of stem-like populations of tumor-initiating cells in glioblastoma multiforme, which happens to be one of the very most aggressive brain tumors. Given that chemotherapeutic drugs, it would be able to kill those cancerous cells before they proliferate again. And that will be done by a lentiviral system that I will be introducing in order to knock down the gene of SPEEDY, and then introduce the chemotherapeutic drugs right after the injection of those cells, those cancerous cells, into an in vivo model called a zebrafish. And by giving those chemotherapeutic drugs, we would be able to see a significance in the decrease of the levels of those cancerous cells. So in conclusion, if we can reach that hypothesis and at least either prove it or disprove it, at least we know from where to take the next step in order for us to find a better treatment for the medulloblastoma instead of having the combination of the radiation, the surgeries, and the chemotherapy, but by just inducing a very decent amount of chemotherapy with the knockdown of that special protein, then maybe we can get with something towards the end.